Good morning, everybody. My name is Christine Durand, and I co-direct the Hopkins CFAR Cure Scientific Working Group, along with Dr. Ashwin Balagopal and Kelly Metcalf-Pate. It's my privilege to welcome you all to our Cure biannual symposium, which we have entitled HIV Cure Just Around the Corner or Miles Away. So the objective of our Cure Scientific Working Group is to nurture a community of investigators from diverse disciplines and at various stages of training. It's really just to get you all into the same room and get you all talking, and we hope by fostering that community that we can spark some progress towards this common goal. Today we have an exciting lineup of speakers, both from inside Hopkins and outside Hopkins, each of whom we believe can lend a unique perspective on the question of cure. We have the community perspective, ethical issues. We're going to talk about some key progress in the areas of basic science, animal models, and clinical research. So we're hoping that you'll all be inspired by these talks, and that during the lunch session and during the poster session, you'll have a chance to network and hopefully inspire one another to new ideas and novel directions for cure research. We're going to end the day with an interactive panel discussion that I'm particularly excited about. Tom Quinn is going to moderate that panel, and we'll also invite a few other Hopkins experts who didn't speak today to serve on that panel and lend their perspective towards this important question. Just a couple of housekeeping items for the day. We are recording these talks, so if the speakers stay on time and we have a few minutes for questions, we're going to ask that you wait to get the microphone so that we can have those questions available to anyone who's listening online. And again, just because we have such an exciting, packed agenda today, we're going to ask the speakers to do their best to stay on time. And if I have to give you a little wave or get you to end, please don't take that personally. We just want to make sure everybody has time to cover their topics. So without further ado, I would like to introduce our first speaker, Lynda D. J.D., who's the Executive Director of AIDS Action Baltimore, and she's going to kick us off today with an exciting presentation entitled Community Call for Cure: Community Engagement, Cure Characteristics, Issues, and Impediments. Thank you, Lynda.
Community person first. I have taught you well. Good morning, everybody. Thanks for having me. This is really very exciting. I love the CFAR. So let's get started so I'm not late. All righty. So this is what I'm going to be talking about, a little bit about AIDS Action Baltimore and our history, myself and Gary Lambert, why we need a cure, characteristics of a cure, ethical, behavioral, social science research issues, and treatment interruptions, which comes under that, which are giving us a huge problem at this point, in my opinion, and how we conduct optimal cure research and funding issues, which are always very important. So we all know about Timothy Ray Brown. This is Timothy, and this is Timothy and his doctor. He's the only person cured of HIV so far, but he's given us the hope to proceed and to at least begin looking at this for a hopefully much wider audience. So this is me, I guess, in 1980. Oh my God, I can't remember. It's supposed to be the men that can't remember. Anyway, I'd like to forget if you ask me, if you want the truth. But anyhow, everybody says to me, well, how did you get into this? And they do it very politely, and I know what they're thinking. So I usually start off with how I got into this. I essentially was a party girl, and that's my husband, and he died from complications of AIDS in 1987. He actually was treated here in 1986. He was in the Osler 5 9 times before he died, okay, with different issues. He had histoplasmosis that nobody knew what it was. They kept saying he had pneumocystis, and he ended up with an exploratory surgery to figure out what's what, and they found this ball of histoplasmosis. He also had KS, which is what finally killed him. But, the 9 times he was there, I saw so many of my friends go through every opportunistic infection, hideous, that you can imagine. So I don't want to dwell on that because it still flashes in front of me when I begin talking about it. But anyway, it wasn't just him, it was all of my friends, and they really, I mean, I lived downtown and worked downtown and went to school downtown, so my gay friends were very good to me, and when it came time to help them, I just felt like I needed to do that. Not only that, after my husband died and so many people died, it really kept my sanity. So it wasn't all altruism. It was like, whew, work, 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 do not think. This is Gary Lambert, who also was one of the co-founders of AIDS Action Baltimore. And the Gary Lambert Research Center was actually named for him. So we were like salt and pepper. I was more fierce, and either you loved me or you hated me, but everybody loved Gary, especially John Bartlett, which turned out to be very important. They were like brothers, so, you know. And actually, I was thinking about this last night. When Gary finally died, unfortunately, days before the cocktail came out, it was John that, I mean, we were all around his bed, and it was John. I said to him, John, you know, he's not going to go until you give him permission. And John whispered something in his ear, and in minutes he was gone. So they were like this, and it was really very helpful to the HIV effort back in those days. We miss you, Gary. Anyway, so why do we need a cure? So we all know about the international pandemic, and every once in a while, I did this as much for myself as for you, because, you know, those big numbers kind of go right over your head, and you don't really think about 1 million people died internationally in 2016. 1 million, total 35 million deaths. 36.7 million people living with HIV worldwide. 1.8 newly diagnosed infections, 5,000 per day. 5,000 people a day. I mean, it's pretty hard to fathom that and to take that in. 160,000 children under age 15, and only 20.9 million are actually accessing antiviral therapy. In the U.S., 1.1 people are living with HIV. 1 in 7 don't know they're infected. I mean, that's really, at this point in time, anyway, it's very unfortunate. Let me just put it that way. There are 39 and 782,000 new infections that were diagnosed in 2016, and MSM are, of course, the highest risk population. So if you're an African American MSM in the United States, you have a 1 in 2 chance, and if you're a Latino MSM, a 1 in 4 chance of becoming infected with HIV in your lifetime. I mean, that's pretty scary. Okay, of course, we all know HIV is an infectious disease. It strikes the young. It's difficult to keep people in care and to keep viral loads suppressed. It presents expensive access and difficult adherence issues. And as far as prevention efforts in the United States, it's much more difficult to engage high-risk minorities in PrEP, and definitely, but last but not least, stigma. So, I mean, I know what I went through, not even having HIV, but having a husband that died from it, with my own family, his own family. Really just, the horror goes on. So, we know about eradication. I mean, I don't want to say HIV is completely eradicated, but, you know, there's a lot of junk there still. So it's non-functional and non-replicating, and you don't need ART. I mean, it's really much more of a clean slate than a functional cure, which you may not need ART, but you might need, and we're talking about this lately, long-acting regimens. You know, if you could if you didn't need to use any drugs for 2 years, 1 year, 5 years, and examples of that are periodic vaccines and the BNABs, that I think people would love and take as a cure. Good enough for now. So we want it to be simple, safe, scalable, and affordable, generalizable in women and men, and for people with chronic, acute infection, and pediatric and adolescent patients. We always forget about those last 2 categories. You know, affordable is extremely important because we talk about monoclonal antibodies that are, you know, historically exponentially more expensive than the drugs that we use now, the small molecules. And, you know, that's only going to be part of the regimen. So we need to be cognizant of that as we go. All righty. So, and this is kind of funny for me because I've been around you guys a long time. And I have a tendency to think, animal studies, basic science, clinical, and then community. And so I have turned it around on its head for this to talk about the community stuff first in all these different categories. And I think that's really important because cure research is going to be different because, initially people hear cure and then, they think cure. When they have any sense and they read the informed consent, I'm not sure I'd want to be the first one in line for these trials. So we're going to need to do a lot of work to make sure we have people that we can accrue after the 10, 20 patient studies when we get really further down the line. And that's going to require work up front rather than at the back end. So we're also talking about nested studies in cure research, like, for example, participant surveys, because we have to know, what do you mean by a cure? What does cure mean to you? No antiviral therapy necessary, long-acting, at what intervals, and inability to transmit HIV, how much risk is tolerable to patients, how much is too much, how many and what types of invasive procedures are they willing to undergo, and how many visits are too many. So, these tend to be, well, I guess we're really only in phase 1 anyway, but they're really, really invasive. And if they're labor intensive for you all, think of what it's like for a patient to be drug in for all of that. So anyway, there's some other concerns too, and we'll call them what you call them therapeutic and curative misconceptions, because people hear cure, and I always wonder, why do people get in phase 1 trials? I know why they did it back in the day when there was nothing for HIV, but why do they do that now? So I think sometimes when people hear the word cure, that's all they hear. They want to be HIV free, and they just think, okay, I'll try this. All right, so we have to be sure that they know what they're getting into, and what terminology should we use. Cure, maybe that's too loaded. Remission, the community doesn't like that word. I think it's very, very medical to them and reminds them of cancer. Durable suppression, I'm not sure. I guess it's going to be patient by patient, but we have to be really careful when we use the word cure. The informed consent process, I think, is broken, and it really is like, how they say about the VA, it depends which VA you're at. This is also site and personnel dependent. Is it truly informed? Do we need these long forms to make sure we cover all the topics? As a lawyer, after page 2 or 3, people just sign. I know that. It's a good thing I'm honest, right? Anyway, the short forms, are they too short? Should we have post-review testing? Do we need to be worried about reimbursement and coercion issues? Probably we always need to be worried about that. And I'm just giving kind of highlights here. So one of the real ethical problems that is, I think, for me, the most serious, these analytic treatment interruptions. We have no way to actually measure, no validated way, let's say that, to measure the latent reservoir, which is the culprit here, because we can kill what's out there and alive and well, but it's this latent reservoir that's the problem. That certainly, where did he go? Dr. Silicano. Bob, did I see you? He was here. Oh, there he is. He taught us about so many years ago. So anyway, they present serious ethical issues. And first of all, we don't need to, this is really very hard to think about our duty of confidentiality to the patients versus partner notification and prevention issues. So if people go on these treatment interruptions, they have a blip and they infect somebody. Some states have criminalization issues. Plus, we just don't want to infect anybody. That's the opposite of what we want to do. So, some of the issues around that are more than one partner. Should we offer the participant counseling, testing, PrEP, PEP, condoms, referral information, or should we get the partner in there to tell them these same sorts, to give them the same sort of advice? And, what do we do when there's more than one partner? Should we provide free PrEP, to the partners? So this is, can you imagine all the things we have to do with these trials, and now we have to worry about PrEP and counseling and whatever. And, there's a lot of legal issues there as well. Once you start giving people advice, all of a sudden you're responsible for what happens to them. So, in other words, I don't know the answer to that at this point. So, as far as these treatment interruptions are concerned, we had a great workshop in Boston in July, headed off by the Reagan Institute, and really Boris Yolg is the ATI hero of this. And we try to come to some consensus. It was actually, we're doing the paper now, the manuscript, almost halfway through the manuscript, I guess. We have to send it out, you know how that goes. But this is kind of what, I'm going to give you the highlights of the problems and issues with this. Age, 18 to 65, or people that are older than 65 too old to stop their therapy. Remember, I mean, the guidelines recommend that you start treatment and start it early. So we're now saying to people, okay, we're going to take you off and see what happens to you. Yeah, sure. So there's a lot of people that think including some people in the community, think that it should be 65 and older, 70, and some people say, if I'm 80, I'm doing fine. If I'm otherwise healthy and not excluded by the criteria, then why not? So we actually had panels and questions and a little bit of voting at the end. So the community was really, I mean, I've been like a maniac with this, and those of you that know me know what that means. That we should not have people in these trials initially, at least, that have less than 500 CD4s to start. So all the docs wanted 350 because, they're used to getting these same old white guys in the trial, and they're a little bit more beat up, and they've been around a little longer, and they have less than 500 CD4s usually. So won that argument. Lost the NADA argument. The community wanted 350, and they actually voted for 200, but I did talk Steve Deeks into using 350 for his first trial. So he's got a big trial coming up called JAWS with like 5 different interventions. So what he does, a lot of people will follow, I'm sure. The exclusion criteria, I'm not going to go over all of it, but the thorny stuff is, let's start at the bottom. The community thought, if you had a stroke, you don't need to be in these trials. And we've gotten some argument with that. The other thing is the cardiovascular disease. If you have a history of it, we'd like for patients to have either EKGs or stress tests to make sure they're good enough shape, to actually stop their therapy for sometimes, 2 months. That's a long time. We're talking about, that's a heart attack issue, okay? That's, as I always say, serious as a heart attack. The other one is if people have active or recent malignancies, what's far enough out? 36 months, 10 years? Same with AIDS-defining conditions. So you can see the kind of issues that we're talking about. This one is really hard. The restart criteria, confirmed CD4 decline to 350, or should we use the percentage? I think most docs would tell you, or researchers would tell you percentage at this point. So these are the criteria. Steve is using all of these but number 4. And number 4 is what people voted on, which was kind of a shocker because everybody thinks that you have to get, the point of these trials is to make somebody an elite controller. So you have to get that viral load up, to get it down, to get it up, to get it down, till you get to the point of being able to control the virus yourself. So I'm not sure you can do that with 1,000, but this reminds me of the CD4, CD4 criteria from back in the day. What numbers should we use? We're like shooting in the dark with this stuff at this point, especially this aspect of it. And, how often should we drag patients in? Once a week? And I think the consensus is for the first week, 8 to 12 weeks, that, once a week is enough. We've started it 3 times, 2 times. We want to be sure to get somebody to know what's going on with their viral load as soon as possible. And bi-weekly thereafter, as long as there's no viral rebound. I think the new area coming up in this is going to be home testing. And the question for that is are all patients going to be able to do that? Maybe some will, maybe some won't. And that might be the safest way to do this. And the most convenient, which means you won't have people dropping out and they will be adherent to the protocol if if we give them a little bit of education on what they need to do. All right, and so as I said before, we need to do community engagement as soon as possible, including advocacy for funding. That's really, we all know from that. I don't have to spend 1 more minute explaining that to you, I don't think. We need to be sure that we are working on accrual and retention issues, and we need to outreach Do we need outreach and education to all communities, and especially communities of color who always get left behind? So wouldn't it be nice if we were all on equal footing at this point and they were right in line with the white guys who just seem to know this information more quickly? That's really pretty much what it is. I mean, not that there aren't other issues about, going, but the white guys get there first. And we need participation in local community cure forums. We've had Debbie come, Bob come, Christine sent somebody from her shop. It's really been great. We at AIDS Action Baltimore once a month have an HIV education session, and I mean, I think they found it useful, and we've definitely found it useful. So, that there are people that you're reaching out to when you need people in your trials, then they're going to think, oh, I know about this. Maybe I'll look into it. They've got to know about it and understand, at least have some semblance of understanding what's up. And again, inclusion of all communities, including, pediatrics. And I thought it was very interesting. Debbie and I were at an NIH meeting last week, and she talked about what a boost it's been for pediatric research, this cure research. I think kind of put you guys back on the map, right? So I think that's very useful. And community advisory boards can be useful reviewing protocols and informed consent forms. I mean, in other words, if there's like this glaring thing that a community person might see that you might not, it would be helpful to have to be able to fix it now before you have to do a protocol amendment when you have nobody coming. Okay, so I mean, here's some of the issues in the organizations. So this is really fostered amazing national and international collaborations. We have the Martin Delaney Collaboratory that has 6 sites now. We used to have 3. And I've actually worked with DARE and CARE, also with AMFAR. The ACTG is doing some very interesting cure research. God, that's where I learned all this stuff back in the day about clinical trials and how they work. That was when I first was with the ACTG, I was on the Opportunistic Infection Committee. That's how long ago. And of course the CFARs, which you have a great one here. Anyway, so we're talking about basic science, animal research, clinical research, ethics, behavioral, and social science issues. I mean, many of these areas have different goals and objectives, and historically they've been funding rivals. So how do we mesh all this together? You'd actually be surprised if you go to one of these cure research forums. I noticed this when the collaboratories first started about 7 years ago. When you see the acknowledgement slide, the groups of people that are collaborating, I've never seen anything like it. It's really pretty amazing. They know they need each other, I think, right? So I think most of you, and I'm not sure of who knows what in this audience, so I figured I better put this in. Here are the strategies that we're looking at. Latency reversing agents, I think Bob coined that term. The kick and kill approach, wake it up and kill it. Get this latent virus and wake it up and kill it. I like block and lock, which means why wake it up and give it all these, give the patient all these horrible drugs? Why not keep it asleep? Keep it there where it is. There's a new agent called DCA for that. I think the problem with this is it something that's going to have to be given every day, very frequently? So that kind of throws the idea of a cure out the window. But I think it's very interesting. Immunomodulators, for instance, CAR T cells, broadly neutralizing antibodies. There's 2 examples of the 2 that are very prominent right now. And gene therapy, the zinc finger and the CRISPR-Cas technology. So I don't need to go into that anymore, but just so you know. But our biggest scientific issue, I think, is what I started with, no validated assay to measure latent virus. Just imagine what it would be like to do an HIV trial without viral load testing at your beck and call. It's just impossible. And I always say, this reservoir, every minute it gets bigger. I always say that every time Bob opens his mouth, the reservoir gets bigger. And I think we all pretty much know now it's everywhere. So, I wish you guys would get this problem solved so we could get rid of these ATIs. And there's also some really neat technology that a doc that I know and love in California is working on, scanning the body to see where it is, to see exactly what compartments it's in, how many compartments, how much of it. So that's really interesting too. You better get on with those assays or he's going to beat you. Anyway, what I say to people is that we're in the AZT phase of this, and people kind of understand that, at least in the community. We're in the beginning, we got a long way to go. And all we really know at this point is that we're gonna need combination therapies. You know, the animal research is ongoing, we're doing that so we can hopefully be more safe when we get to humans, but of course there's always the age-old problem, will those findings translate to humans? We have regulatory issues. I mean, a really huge one is the FDA classifies asymptomatic people with HIV as healthy volunteers. So that risk-benefit ratio is really a tough thing to overcome when you're talking about dangerous trials. You know, you have people that aren't sick and that have the option of a number of once-a-day regimens. So how do you mesh that? You get a community person to talk to the FDA for you. I think that's what's been happening. And they also are insisting on mitigating this kick and kill strategy that has, that the kill aspect of it are cancer-causing drugs. And, you know, a lot of the drugs that they started with in the beginning were used in cancer research. And what they have made people do is do dose ranging, small N studies, and kind of phase in participants, making sure, you know, we don't hurt anybody initially. And finally, funding. So these are figures from the OAR professional judgment budget for 2019. Now, I'm not going to give you 2018 because that's probably what we're going to get, you know, hopefully not. We're kind of working on that. But I just want to give you an idea of what the OAR thinks is important as far as numbers are concerned. That's probably not fair. What, you know, there's scientific opportunity and all that sort of thing. But anyway, they've asked for a 15% increase in the professional judgment budget for 2019. So it's an increase of 14% for reducing HIV incidence and 1% for next generation therapies. I don't know why industry doesn't do all that. I'm not sure why we have to give that anything, but that's just my opinion. You know, cure, 157 million, an increase of 3.5. Like, you know, it's the second lowest one. And if you ask Tony, it's because of maybe there's more scientific opportunity in other areas. Comorbidities and co-infections involved with HIV, 17.6. Cross-cutting basic health disparities, training, behavioral and social science, 19.6. I wonder how much of that 19.6, if we get it, will actually go to behavioral and social science. But anyway, something that we'll have to keep on them about. So again, you know, when you think about the actual impediments that we have, I mean, I guess that goes hand in hand as what's commensurate with scientific opportunity, but we're not, you know, we're second to last on this. It's really not very encouraging. And I have to say that I was a bit discouraged when all of the kick and kill, which was, you know, the most advanced strategy, nothing really worked. So it was kind of depressing and discouraging. But as I said, we were at this meeting last week, and there was really a lot of work being done in a lot of different areas, especially in the immune system. So it was very encouraging to see that you know, the wheels were still turning. So I think as a result of this, even if we get a 3.5 increase, we're going to have to make the best of, make the most of basic science and translational funding and work together with those researchers and make the most of the Martin Delaney Collaboratory, the ACTG, and things like your CFAR. I think, again, this has really fostered unique and amazing collaborations, and it's, some of it's evidenced by the stellar CFAR that you have at Hopkins. All righty, so this is interesting to me. So we have all the 14% for reducing incidence, and then we have 3.5 for cure research. So for me, that presents yet another ethical quandary, especially for people of color. So we're telling people, we've told them for years, take your drugs, take your drugs, take your drugs. PrEP, got to get in there. If you take it, if you take it, if you take it, it will work. If you take it. And now we have you versus you. You know, undetectable means untransmittable, which essentially, if you're undetectable, you know, I mean, God, when they put all those studies together, you'd have to have sex for 400 years to infect somebody. So that's pretty good, right? Anyway, but when you think about the implementation funding for all that as well, when you think about prevention versus cure, and that take your drugs now, we're taking you off drugs, it really fosters medical mistrust in the African American community and communities of color, because we're really kind of reaching out to them to try and get them engaged in the PrEP and the prevention world, and now we're saying stop your drugs. So they don't trust anything we say. So you just need to be advised of what a non sequitur that is for them. So plenty of issues to get to work on here, and I'd like to thank the CFAR for having me. I'm very loyal to Hopkins. As I said, my husband was here for a very long time, and I watched, and I'm talking about 1986. I mean, that's pretty good to have a whole dedicated unit to HIV and to be working on it all these years after, so thank you for that. I want to thank Dick Chason and Chris. I mean, Dick is the glue with this. I mean, with the work that he's done with this CFAR, it's really been amazing. Yay. Anyway, thank you, Christine, for asking me. I hope this is what you wanted. Thank you, Debbie and Janet and Bob, for coming to see us at AIDS Action Baltimore and our community forums. If you're interested, I'd love to have any of you come and talk to our group. Even if you want, people for your trials, we're happy to do that. Ann and Eileen, because we really know who runs the show, don't we? And all the trial participants and volunteers that help you all get to where you need to be. So, thanks. 1 minute to spare, so I guess you want to ask questions later, or?
Yeah,
I think, okay.
You were right on time. Perfect. Stay on.
Well, that was outstanding and a great way to summarize not just the community and ethical principles involved in a cure, but just even the day, setting the stage for what's to come. It's my privilege to introduce Dr. Blankson, who earned his MD from Cornell University and his PhD from Rockefeller. He did his internship, residency, and infectious diseases fellowship here at Johns Hopkins and worked with Dr. Silicano on the latent reservoir and acute seroconverters. He's a professor in the Department of Medicine and in molecular and comparative path, and he serves as the director of the inpatient HIV unit. He's known for his work on elite control in HIV and HIV cure strategies. It's a real privilege to introduce Dr. Joel Blankson. Thank you.
Thank you, Ashwin, for that kind introduction. I want to thank the organizers for inviting me to give this talk. So I'm going to give you a story in 3 different acts. Okay. So unfortunately, I don't have any financial disclosures to tell you about. It's pretty sad. So you heard about HIV cure, the 2 types, the physical cure and the functional cure remission. And physical cure is complete eradication of all replication-competent virus, whereas a functional cure is when you have some remaining virus that you control without ART. So a physical cure is like when you get the flu, it's completely eradicated. A functional cure is any herpes virus. It's with you forever. So we only have 1 possible case of a physical cure. So it's not 1 in a million, it's 1 in 37 million. That's the Berlin patient. Whereas for functional cures, we have HIV controllers, we have the Mississippi infant, and the Boston patients. So not a lot of patients, but at least it's much more than the physical cure, and it's probably much more achievable. So Act 1 of this story started in the late 1990s, and it started around about midnight. And this is one of my favorite CDs by Miles. Okay. So a middle-aged man presented with acute delirium, and he was later found to be unresponsive. He was rushed to the emergency room, admitted to intensive care. So he had a CT scan that showed no abnormalities, so he got a spinal tap. And the spinal tap showed white blood cells, mostly mononuclear cells, elevated protein, which was suggestive of an infection, but his gram stain was negative and the HSV PCR was negative. And it was a bit of a dilemma until somebody got an HIV antibody test, which came back indeterminate, and his viral load was really, really high. which is very suggestive of primary infection. So the diagnosis was meningoencephalitis from primary HIV infection. So it turned out Justin MacArthur was the neurology attending at the time, and he, with the ID physicians, recommended the patient go on antiretroviral therapy right away. And he improved markedly, and he walked out of the hospital with no deficits in a few days. So this is his clinical course. In red is his viral load, blue is his CD4 count, and as you can see, he had an appropriate response to ART, which is shown in gray. But then he stopped taking it for a while, and his viral load rebounded immediately. He was put back on ART, and again, he suppressed his viral load. But around 2002, he just stopped taking it, taking ART, that is. And he has been undetectable ever since, so that's 16 years. So we call these patients post-treatment controllers, okay? And they're defined as patients who suppress viral replication for prolonged periods when ART is discontinued. So the median time to viral rebound when patients stop ART is typically 17 days. So this is a classic study from 1999, and you can see most patients rebounded right around 20 days, 17 to 20 days, okay? So one definition of post-treatment control is control for more than 24 weeks. It's very arbitrary, obviously. So a recent study found that 13% of patients who are treated during primary infection will become post-treatment controllers. But this is short-term control. If you look at 2 years out, it's only 6.5% of all patients will be post-treatment controllers. And if you get out to 5 years, it's down to 2.6%. So long-term post-treatment control is not common. And if you take a patient who's been out 16 years, that makes him the undisputed heavyweight champion of post-treatment controllers, right? Okay. So there are two types of controllers, right? We talked about post-treatment controllers. There's also elite controllers or elite suppressors. And the difference are elite controllers are patients who have never been on ART, okay? So a question you might be thinking is, well, how do I know this patient wasn't going to become a elite controller? How do I know this is really post-treatment control? And the answer is you can never definitively tell. But statistically, post-treatment control is much more common than elite control. Elite control is less than 1% of all HIV-positive patients. We also know that elite controllers tend to have protective HLA alleles, whereas these alleles are rare in post-treatment controllers. And elite controllers tend to have blunted viral loads in primary infection, whereas post-treatment controllers would typically have the high viral loads early on. But now that we start ART so early, it's becoming harder and harder to distinguish between the two. In terms of our patient, he had a very high viral load and was very symptomatic. And I think he probably would have died without ART. So I don't think he was going to become a post-treatment controller. I think he was going to become a rapid progressor. Okay. So why the fascination of post-treatment controllers? So elite control is thought to be due to genetics. And if you're not elite, nothing can make you elite. Whereas post-treatment control is thought to be achievable by a much higher percentage of patients. Therefore, it's more applicable to the cure agenda. So it's 13% versus 1%. And as somebody once said, early ART can make you a post-treatment controller, whereas only God can make you elite. So I think that's wrong and elitist, but that's just me. So this brings up Act 2, which happened around 2010, and it's named Mysterioso for Thelonious Monk. Okay. So the star of this act is Maria Salgado, who was a very talented postdoc from Madrid. And this is her supporting cast. So Maria asked, what's the mechanism behind the control? So he did not have any protective HLA alleles, and we ruled out the fact that he might be taking ART on the sly by doing mass spec of his plasma, don't know ARVs. So Maria looked at his replication competent virus. She went into his reservoir, pulled out some virus, and as you can see, in blue is his virus from 1999, red is his virus from 2010, and those viruses replicated just as well as lab strains, BAL and 3B. Okay, so fully replication competent. Now, patients on ART have a reservoir where the frequency is about 1 infected cell per million. We know elite suppressors have much lower numbers, anywhere from 10 to 100-fold lower. Well, this patient, patient 169, was different in that his reservoir was much bigger than elite controllers. It was like the average patient on ART. So that's impressive. Furthermore, it turns out patient 169's spouse transmitted the virus to him. And this is phylogenetic analysis of envelope. And his spouse, who had a CD4 count less than 200 and high viral load, her virus was much more closely related to his than any other virus in the database. So this is a transmission pair. And if she had progressive disease, it looks like it's not the virus, but the patient. So it turns out the patient also had dual tropic virus, which is unusual because in primary infection, it's normally R5 virus that's transmitted. But if you look at this virus, it could infect, shown here in the box, it could infect cells that expressed only CCR5 or CXCR4 or both. Okay. So Maria then asked, could this be that his CD4 T cells were resistant to infection? So he had normal CCR5 levels, and she looked at infection by both X4 virus and R5 virus. So these are healthy donors, and this is the patient. And as you can see, his cells were very infectable by both viruses, so cells were not resistant. So I told you about the class 1 alleles, the protective class 1 alleles you see in elite control. It's usually HLA-B57 and B27, and they're usually just reflective of a good T cell response. It's thought that those alleles present conserved peptides to CD8 T cells. So we asked whether this patient had a good CD8 T cell response in the absence of the alleles. And we do a suppression assay where we take CD4 T cells, we infect them with HIV, you get infected CD4 T cells, and then you add CD8 T cells to one arm. And if you have good T cell function, you should see much less virus released compared to the control arm. Okay? So when we looked at HIV negative donors, so red is CD4 alone, green is CD4 with CD8s. So these are negative control, and you can see the CD8s did nothing. If you look at elite suppressors, the CD8s do a really good job of suppressing virus. Whereas with patient 169, his CD8s did nothing at all. So then we looked at his neutralizing antibody titers to see if it was antibody-mediated control. And elites in general have low titers of neutralizing antibodies to lab strains compared to viremic patients. And patient 169 also had a low titer. So Justin Bailey then looked to see whether he had antibodies to his own envelope. So this dashed line is envelope from 2010. And as you can see, his antibodies were not able to suppress his own envelope, so he didn't have high titers of neutralizing antibodies. So this is a gentleman who had severe encephalitis and a high viral load. He maintained undetectable viral loads off ART for 9 years at that point. He did not have protective HLA alleles, and he had replication-competent virus. But what we thought was really special was the fact that he had a huge reservoir, unlike elite suppressors, and he had unimpressive HIV-specific T cell responses. So we thought whatever the mechanism of control was, it would be more applicable or easier to achieve than that of elites. And I thought this was more impressive than elite control because the reservoir is so much larger than what you have with elites. So around that time, the Visconti cohort started to get a lot of press. The Visconti cohort is in Paris, 14 patients who were post-treatment controllers that looked just like ours. And like our patients, they had very few protective HLA alleles, and they had weak T cell responses. The difference being that they had small reservoirs, whereas ours had a large reservoir. And they had no mechanism. So we couldn't find the mechanism, so we went ahead and published our paper. I wasn't happy about it, but that's life. But that led to Act 3, which was in 2017 to 2018. And this is called Giant Steps. So, after John Coltrane. So, again, I was very disappointed that we never found a mechanism. And we kept trying, and we wouldn't get very far. But in 2017, we got a really talented postdoc. And I flat out lied to her and told her this was an easy project and she'd have a paper in 2 weeks. And she jumped on board. So this is our supporting cast. So 2017, she attempted to culture virus from 2 million resting CD4 T cells, and we got nothing, which was very frustrating, but it turned out to be a blessing in disguise. So Rebecca said, just bring him back, let's do it again. So we did a lot more CD4 T cells, and this time we got 7 isolates. Of the 7, 5 had identical sequences. So we brought him back again, and we got 4 more isolates, and 4 had identical sequence. So 9 out of 12 of replication-competent virus had identical full-length sequence, okay, so you're wondering, why do I keep underlining the word identical? And this is the HIV life cycle, and one of the most unique steps is reverse transcription. It's where you go from RNA to DNA. Now, reverse transcriptase has a high error rate, 1 per 30,000, 1 mutation per 30,000 nucleotides, which means 1 in 3 infected cells should have a mutation after 1 round of replication. And as you have exponential rounds of replication, you should see more and more mutations. In contrast, DNA polymerase has an error rate that's 1 per billion. So when you start seeing a lot of identical sequences, it suggests that you're not having viral replication, but you're amplifying the virus by some other source. So 2 methods of virus expansion. One is virus replication, where you have an infected activated CD4 T cell. It's going to make virions, and that's going to infect uninfected activated CD4 T cells. Okay. So even if you have the same virus starting out, because of the error rate of reverse transcriptase, the infected cells are going to be a little bit different, right? Because of the error rate, there's going to be some mutations. And therefore, when a virus is produced, you're going to see a virus that is very similar, but not identical. All right. Now, these activated infected cells lead to latently infected CD4 T cells. And this is the main reason why we can't cure HIV. These cells are quiescent. The virus is not replicating, so ART has no effect. The cells are not expressing proteins, so the immune system can't clear them. It's not the only reservoir. Myeloid cells also are a reservoir, and you'll probably hear more about that today, but this is a major reservoir. And the problem turns out to be a little bit harder than we thought it was, because it turns out you can have clonal expansion of some clones. Okay, so like the red clone will expand, which might be due to homeostatic proliferation. And if one of these cells become activated, you're going to produce virus which will be identical. Okay, so that's how you distinguish between clonal expansion and viral replication. With viral replication, you have sequences that are similar but not identical, whereas with clonal expansion, the sequence should be identical. So this degree of clonal expansion, it was unusual. And if you compare it to chronically infected patients on ART, this is some of the work from Bob's lab. You see clonal expansion, but you also see diversity suggestive of viral replication. Okay. So we asked, could it just be because we started ART early in this patient that he had all this identical sequence? And Matthias Lichterfeld looked at 2 patients who had primary infection, where he did full-length sequencing, and he did not see a lot of identical sequences. He saw a lot of homogeneous isolates, but they weren't identical. So another way you can look at this is by looking at integration sites. Integration is a random event. It's virus gets preferably integrated into actively transcribed genes, but the exact site should be different. So if you're trying to differentiate between one dominant clone early on that's seeded many different cells versus clonal expansion, in the former, you should see integration at many different sites, whereas in the latter, there should be one integration site. So Rick Bushman at Penn helped us with this. He has this method, which I will not go into. But what we did see was that there was repeated integration sites, suggesting that there was some clonal expansion. Now, what we have not been able to prove is that the integration sites correlated to the expanded sequences, or that the integration sites represented replication competent sequence, and we're working on that. So we have strong but not definitive evidence of clonal expansion in this patient. But what's the mechanism of control? So going back to the suppression assay, Rebecca said, let's do it again, but let's use the clonal virus to see if there's a difference. So this is the suppression assay, and in blue is an elite controller, and you can see his CD8 T cells suppressed very nicely. In red is patient 169, and the CD8 T cells does absolutely nothing, okay? So then we got the autologous clonal virus and looked at suppression. And surprisingly, patient 169 CD8 T cells suppressed its own virus just as well as the elite controller CD8 T cells controlled their own virus, okay? In contrast, patient 169's spouse, her CD8 T cells did not control her own virus, okay? So we asked, what's different about the autologous virus? Could it be just that it's not very fit, so it's easy to suppress? So we looked at viral fitness, and in red is patient 169's virus. It's just as fit. This is the clonal virus. It's just as fit as lab strains and as his spouse's virus. So then we asked, could it be due to difference in HLA downregulation, which is a common mechanism of immune evasion. So what the NEF protein does is it will pull down MHC from the cell surface, so CD8 T cells do not see infected cells. So here we're measuring HLA on the cell surface. This is an infected cell, and I'm sorry, this is an uninfected cell, and these are infected cells, and you can see there's downregulation of HLA. But his virus shown here downregulates HLA just as well as the lab strains and his spouse's virus, so it wasn't that. So we looked at the epitopes, so the part of the protein that the CD8 T cells targeted, asking whether those differences between the lab strain and his own virus. And we didn't find any major differences. And there wasn't a lot of evolution between 1999 and 2017. Okay. We looked at GAG and NEF and didn't see a lot of difference. So we saw strong evidence of clonal expansion. We showed that the reservoir did not expand over 6 years, and we showed that his CD8 T cells could inhibit his own autologous clonal virus. So as Bob would say, so what? Well, we talked about the two types of viral expansions, viral replication and clonal expansion. Now, viral replication will be blocked by ART, but clonal expansion, the viral genome is being copied by human DNA polymerase, so it's not going to be affected by ART. So technically, you could have this expanding even while you're on ART. What about CD8 T cells? We think elite controllers, CD8 T cells, inhibit viral replication, but this should not be inhibited by CD8 T cells because these cells are not making proteins. Okay, and we looked at this by looking at the reservoir of elite controllers versus patients on ART and viremic patients. And we looked at the frequency of identical sequences. We hypothesized that if CD8 T cells could wipe out virus produced by viral replication, you'd see a larger percentage of virus that was due to clonal expansion. And that's what we saw. We saw much more clonal expansion in elites compared to patients on ART and viremic patients. So while this could be a problem, clonal expansion, because it's not inhibited by ART or CD8 T cells, in order to get viremia, the virus that's produced has to infect other CD4 T cells, okay? And when that happens, CTL or CD8 T cells can control it at that point. So we think HIV-specific CD8 T cells might not be able to prevent clonal expansion, which might be a problem for the cure agenda. But if this virus goes into other CD4 T cells, CD8 T cells can wipe them out at that point. And we can have viral remission despite clonal expansion of replication-competent virus. So while clonal expansion may be a barrier to physical eradication, it doesn't have to be a barrier to a functional cure. So hopefully I'll tell you about Act 4 the next time we have a symposium. And these are some of the questions we want to address, and we're working on this as we speak. But going back to HIV cure, personally, I think a functional cure is much more likely to happen than a physical cure. And I think if you got patients really early on, put them on ART, had the immune system preserved, and then immunized them, those patients would be most likely to achieve a functional cure. So I want to thank Maria Salgado, Ali, Karen, Bobby, Justin, who did all the work. Joe Marglick enrolled a patient in his study in 1999. Bob's two mentors. 2017, Rebecca Wienheis did most of the work with Abner, Caroline, Rachel, Chris, Eileen, and Justin. Rick and Chris did the integration site analysis. I want to thank my funders at NIAID and CFAR, and I really want to thank Ashwin, Kelly, and Christine for putting on this fantastic symposium. And most of all, I want to thank patient 169. So he's a post-treatment controller, but in my estimation, he's always been elite. So, thank you.
I think we have a few minutes for questions before the next session. If you'll just remember to use the microphone so that we can record some of the questions.
Hi, Bob.
Thanks, Joel, for a beautiful talk. Is it possible that he has a very strong CD8 response to a previously unidentified epitope restricted by one of his perhaps less studied MHC alleles, and that it's an epitope that is, one of these things that Bruce Walker talks about being highly networked that basically can't change without a loss in fitness?
Yeah, it's possible. We did blind screening with overlapping peptides. So we didn't look at predetermined epitopes. We just looked at overlapping epitopes, overlapping peptides, and we found those responses. I haven't checked to see if there are any that Bruce thinks belong to the networks, but that's a good idea to look at. Yeah.
You were looking at clonality essentially by looking at the sequence of the virus. Did you look at T-cell receptor clonality?
No, we want to do that. It's challenging to do because you have to isolate the particular cell first. So we are looking at ways to do that, but that's definitely one of the things we want to look at.
Great work, Joel.
Thank you.
Really nice.
Thank you, Guido.
So one thing to keep in mind is that the mechanism of CD8 control that you described is actually quite compatible with some sort of transcriptional silencing of the virus in the CD4 cells, which would explain, the lack of effect in expanding, the clones of CD4 cells that are infected, and the lack of evidence that there is a particularly strong CTR response, that would lead to a decline of the reservoir size, over time. So I just think it's fascinating, and I think it's important that we keep in mind that those alternative modes of actions of CD8 T cells when examining this data. I mean, this patient I think it's really informative in its sort of peculiar phenotype, and hopefully will make people think about what the mechanism could be. Not just the classical, killing.
Sure. So we did show it was contact dependent and was class 1 dependent, but we haven't gone further than that. And I agree with you. It'll be interesting to do that. Hi, Kate.
Hey, that was a great talk. I was interested by the fact that, and I don't remember the exact timing, but when your patient initially stopped antiretrovirals, he rebounded quite quickly, and then went back on antiretrovirals for some period of time. I wonder if you could comment on how that plays into what happened exactly. If this is CD8-mediated control or immune-mediated control, why didn't it happen the first time, and then why? I mean, he doesn't even have any blips from that point on. It's just complete suppression after that initial rebound.
Yeah, it's a fantastic question, and The second time he stopped ART, he did it by himself. Nobody knew. And he just showed up a few years later. It was undetectable. And had it been in an ATI trial, we probably would have started ART right away and not known. So it's a great question. I think if we had left him alone in 2002, his viral load would have come down by itself. The fact that it went up but came down again is suggestive of immune-mediated control, especially since his reservoir is so large. I don't think it was because we decreased the size of the reservoir. So I wish we had known when he had initially stopped the second time around to follow that.
I think the first question is the related question. Did the coronavirus emerge during the first interruption?
Yeah, unfortunately, we don't have that plasma.
Oh.
Unfortunately, that's something we would like to have had.
And the second question is, did you also look at the neutralization antibody, but did you look at the binding antibody as well?
No, we haven't looked at that. That's a good question. Yeah.
Joel, I have a quick question. Your autologous suppression, the suppression of autologous virus, raises the question as to whether you think he would be protected from a new infection, and what that means pragmatically for post-treatment controllers.
That's a great question. There are case reports of elite controllers who become super infected and crash and burn. There are other case reports of elite controllers who are infected with multiple viruses and maintain control. I think the fact that his response appears to be narrow would suggest that he may not protect. And we're trying to model it in mice, but we haven't gotten there yet. It's a great question. Thank you.
Thanks so much. That was a fantastic talk and question session. It really helped to frame some of the HIV biology questions. It's my pleasure to introduce Dr. Shan, who I met when he was doing his postdoc with Dr. Silicano. He did his PhD with Dr. Silicano, and there he focused on in vitro models of HIV latency and virus-specific CD8 T-cell responses. He joined Dr. Rich Flavel's lab at Yale to develop humanized mouse models to study the human immune system. He's most recently joined Washington University in September of 2017, where he's using humanized mice to study how immune systems respond to HIV. And with that introduction, it's my pleasure to introduce Dr. Liang Shan.
Thanks for the kind introduction, and thanks for having me here. I feel great to be here, see all the very familiar faces and the familiar environment. I haven't been here for a while, so I'm glad to be back here. Today I'm going to introduce some of the recently developed humanized mouse models in the course of my study, and I think it could be very useful to study immune clearance of the viral reservoir. Luckily, I don't have to introduce a lot of HIV latency background thanks to the decades of brilliant work by the Silicano group, as well as other HIV/AIDS community at Johns Hopkins Medical Campus. So briefly, HIV infection can persist in long-lived host cells, primarily resting memory CD4 T cells. Within the host cells, the virus is in the integrated proviral form without detectable viral gene transcription, make it invisible to immune system. Because the long half-life of resting memory CD4 T cells, so to allow this reservoir, which is about 1 to 10 million cells per patient, to fully decay, it takes about 60 to 70 years. Therefore, once the patient is infected, they will be infected for lifelong and has to remain on treatment for lifelong. Treatment interruption will lead to quick virus rebound. So in order to achieve no matter eradication or functional cure, we have to accelerate the decay rate of the viral reservoir. And step one for the shot and kill strategy is to use small chemical compound to reactivate latent HIV to make virus to start to produce viral gene products. and at the same time avoid inducing global T cell activation. And with the hope that viral gene transcription or protein production will induce the cell death. And for all the shocking kills that use latency reversal agent to treat HIV patient, although cell-associated viral RNA can be detected, those trials failed to detect clearance of viral reservoir or even reduction in HIV DNA frequency in the blood. So during my PhD study in Bob's lab, we found that in a latency model, in vitro latency model, that our treated patient did not have functional CD8 T cells to clear the reservoir cells in which HIV gene transcription has been reactivated. We have to stimulate a stronger CTL response to clear the reservoir. But this is still far from the truth, because in clinical trials in which functional CD8 T cell response had been induced, there was no clear clinical benefit in terms of delay of viral rebound or reduction in HIV DNA in the blood. And later on, Kai from Bob's lab identified that was probably because the dominance of CTL escape mutations in vast majority of the latent HIV, which means that the immune-dominant CTL response will not be able to recognize those latent virus, despite being induced by any kind of conventional vaccine strategies. And how about the neutralization antibodies? So we know from previous clinical trials that a single BNAb treatment will not be able to clear reservoir or even suppress viral replication, because all the patient will either have baseline resistance or rapidly develop resistant mutations to the BNAb. And also in a recent study that published using a combination of two BNAbs, only a small fraction of patients will be eligible because they don't have the baseline resistant mutations to neither of the BNAbs. So there's still a long way to go to overcome the barriers to effective immunotherapy. No matter for CD8 T cell response or BNAbs, we have to develop a tool to effectively predict the treatment efficacy. And hopefully we can be able to improve the efficacy. And here comes, I think, a small animal model would be very useful to achieve these goals. So to develop a humanized mouse model for HIV cure research, there are two criteria, I think. One, you have to develop a truly the target cells that fits the bill for latent reservoir, which means long-lived CD4 positive T cells, and perhaps tissue macrophages, which is also long-lived and can be potentially a reservoir as well. And second, you have to develop all the effector cells of the human immune system that includes CD8 T cells, NK cells, monocytes, macrophage. B cells is also important, but in terms of therapeutic antibodies, it can be bypassed. And also, we still don't understand how to induce BNAbs in humans, so that's still a question mark. It's just a brief introduction of all the humanized system. All the system is based on either a skip mutation or a RAG2 mutation, which blocks the TCR and BCR genomic rearrangement so that mouse will not develop either B cells or T cells. And common IL-2 receptor gamma chain knockout further block NK cell development. So it's a knockout of mouse primarily adaptive immune system. And then these can be built on either the NOT background or BOB-C background to achieve the similar outcome. And another fact is the source of human cells. In the late 1980s or early 1990s, human PBMC were primarily used to generate humanized mouse models. The limitation is that you inject human T cells into the mouse that create a very strong GVHD syndrome and cytokine syndrome. So the window for that study is about a few weeks, so a lot of limitation, especially for latency research. And then since the early 2000, more and more labs started to use either fetal liver or cord blood CD34 cells to engraft mice. and hope that these cells can differentiate into multi-lineage human immune cells. And ideally, we want to also be able to use patient bone marrow or mobilized blood CD34 cells so that we can use a personalized medicine approach to look at, for example, CAR T or adaptive transfer T cell therapy or antibody therapy. And one of the models particularly used in HIV research is named BLT, which requires fetal liver CD34 positive cells plus matched thymic and liver tissues. This model is not related to what mice strain, it's related to how we operate the engraftment. And for most common procedure to generate humanized mouse, you take human CD34 cells and engraft into newborn mice with the hope that in 2 or 3 months, it will develop all these different human immune cells, which is not the case. So in conventional humanized mouse, what you develop is primarily B cells and a little bit T cells. But if you look at the human blood, myeloid cells are the dominant subset of the immune cells in human blood. Why is that? It's because important essential cytokines or growth factors are not well conserved between human and mouse, especially for those myelopoiesis required cytokines. So in Richard Lab, about 12 years ago, it started to generate those human cytokine knocking mice. We take there are 2 criteria. First is the cytokines are not crossly reactive. In this map, which means it's either gray or red or yellow, we have low amino acid identity. And second is those growth factors or cytokines cannot be produced by human hematopoietic cells, which means the engrafted human cells cannot compensate for the cytokine production. Or at least it's not the major source of those cytokine production in vivo. So for early hematopoiesis, we humanized the TPO, IL-3, IL-6 genes. And for myelopoiesis, on top of those genes, we further humanized GM-CSF and M-CSF genes. And for lymphocytes development, we humanized IL-7, IL-15, and human MHC class 1 molecule. So how do we do this humanization? This is a big project. We want to keep the entire open reading frame because it related to alternative splicing, and also there are non-coding RNA that can regulate gene transcription translation. So in this case, this is the humanized allele in a mouse that contains the entire human open reading frame, which replaced the mouse one under the control of mouse promoter. So just imagine when we put the human stem cell into the bone marrow niches, the mouse bone marrow stromal cells will produce human cytokines to support their development and differentiation, which is a human knock-in on top of mouse knockout. That really created great niches for human hematopoietic stem cells to grow. So we create all those single knock-in mice strains, and then the first combination we made, we call that Mr. G, which has M-CSF, IL-3, GM-CSF, and knock-ins, which boost the myeloid development, and SIRP-alpha knock-in to help mouse phagocytes develop tolerance to human cells. And TPO knock-in is important for long-term maintenance of functional HSCs. And RAG1 IL-2 receptor gamma chain knockout is very conventional. So 6 years later, we had another version that includes all the cytokines related to lymphocyte development. So this is not published, so we don't have an official name yet, but consider we have 10 human cytokines knocked into a single mouse, we probably can call it Mr. 10. And then this is the current version. And how these mice can do for us. If you look at our first version, Mr. G, you can see a beautiful reconstitution of human myeloid cells which stain CD33 positive in our Mr. G mice, but you don't see that in mice without those cytokine knock-ins. And do these mice bleed like a human? Well, answer is quite well. you have more human myeloid cells developed in Mr. G or Mr. G IL-6, IL-15 knocking mice. You have better NK cells, and you have lower T cells and B cells. It's not that they're not important. They're important, but we want to recapitulate human blood cell composition better. And with all the early hematopoietic factors humanized, we are able to use as low as 1,000 human CD34 cells and put them into a mouse. We still develop human immune system in a mouse. And the composition is actually very comparable between the high dose, which is 100,000 cells, or low dose, 1,000 cells. You still see bone marrow development of all lineage cells, and then a reconstitution of human T cells, B cells, myeloid cells, NK cells in different tissues. So this is just a summary slides that compare different strain of mice with low dose. For example, 3,000 fetal liver CD34 or 5,000 cord blood CD34. And you can see that with low dose, NSG mice barely reconstitute any human cells, whereas it's more than enough for our new Mr. G6 mice or Mr. G mice. What's more important is that given the better niche to support human hematopoiesis, we want to test if patient CD34 cells can also develop. We know that fetal liver or cord blood CD34 cells have greater potential of differentiation, and it's not true for patient CD34. So we used to attempt to engraft patient CD34 cells in NSG mice, and you can see that even you put 200 or 500,000 cells into an NSG mouse, they don't grow any human CD45 cell immune cells out of the mouse. But with our model, no matter Mr. G or Mr. G6, which is better, we are able to help adult CD34 cells to differentiate into human immune cells. So if we look at the tissue, which is probably more relevant to the viral reservoir, we know that monocytes are short-lived. They either die or differentiate into tissue macrophages. And tissue macrophages are long-lived and could be a potential reservoir. So our Mr. G mice with the differentiation factors, including MCSF and GM-CSF humanized, we can see nice reconstitution of tissue macrophages, which stain with human CD68 in lung, liver, colon. These are non-lymphoid tissues, whereas lack of the cytokine, NSG mice cannot reconstitute those cells. And how about the blood monocytes? Are they functioning? So there are three subsets of monocytes in human blood. We define by CD14 and CD16 expression. And these number 1 and number 2 fractions are more phagocytic active. And we can see our reconstitution, the reconstitution of three subsets of monocytes in the Mr. G blood compared to human blood. And they're functioning. They can phagocytose GFP-labeled E. coli very efficiently, whereas the third population is more of a patrolling macrophage that secrete cytokines, have more regulatory functions. And whether they can be active in terms of eating HIV-infected cells. So this is a in vitro study. We take CD4 T cells from engrafted Mr. G mice, infect them with an NL43, and then 3 days later, we co-culture with monocytes isolated from the same cohort of the Mr. G mice, so they're donor matched. And then with antibody, in this case, we use the PGT121, we can see that monocytes can kill more infected CD4 T cells more efficiently. And if we label the target cells with CFSE, we can see more CFSE positive monocytes in the culture with the presence of broadly neutralization antibody. So to study the T cell reservoir, one thing that's extremely important is the reservoir has to be stable. In humanized mouse, apparently we don't have the cytokine to support homostatic proliferation and long-term survival of CD4 positive T cells, which is totally IL-7 dependent and which is not cross-reactive between human and mouse. So with the human IL-7 knock-in model, we did a very simple experiment. We take T cells, in this case memory CD4 T cells, from Mr. G6 IL-6 knock-in mice, and transfer to recipient mice, which is either Mr. G6 or Mr. G6 plus IL-7 knock-in. And you can see 4 weeks after T cell transfer, you see significantly more remaining CD4 T cells in a mouse that produce human IL-7. And what's more interesting is in Mr. G6 mice, you can see that vast majority of the remaining CD4 T cells are those relatively short-lived effector memory CD4 T cells, whereas in IL-7 knock-in model, you can see two nice population of both effector and central memory CD4 T cells, separated by CCR7 staining. So next, we also want to, since both IL-7 and IL-15 are important in T cell homeostasis, so we also made an IL-15 knock-in. And IL-15 knock-in, the first phenotype and the most striking phenotype is on NK cells, because NK cell development starting from bone marrow stage is IL-15 dependent. And in this model, we didn't use any Mr. G background. We just used a very simple background, an SG or SRG, which is conventional humanized mouse model. But on top of that, we humanized IL-15 locus. And in IL-15 humanization mouse model, you see more NKP46 positive cells, which are NK cells. And the NK cell blood count in our SRG15 mice is around 400 to 500 cells per microliter blood, which falls well in the range of human blood NK counts. And what's more important is not about how many cells are they, it's about whether they're functioning, whether they're matured. So in human blood, there are two subsets of NK cells. One is cytokine producing, one is cytolytic NK, and separate by CD56 and 16 staining. So the 56 bright, 16 active are the cytokine producing NK, and these 56 dim, CD16 positive cells are the cytolytic NK, which is more relevant to the cure research because you need the CD16, which is the antibody Fc receptor, to recognize the infected cells coded by antibody. And in our SRG15 mice, this is the only mouse model that can actually develop the more matured NK cell subset, which is ADCC active. And actually, it's very similar to what we can see from human blood samples between our mice and human. We also did a mass cytometry analysis, which includes 33 parameters, mainly focused on activating or inhibitory receptors on NK cells to see whether the regulation of NK cell function can mimic what's happening in human. And so each dot represents a single cell event. Its location is determined by the 33 parameters as a whole. And the color represents the expression level of the particular cytokine, in this case CD94. So we can see that between SRG15 mice and human blood, the NK cell subsets are very similar in terms of distribution and gene expression pattern. So to validate the functionality of our NK cells, in this case, I didn't do HIV research, but I did a tumor study. So it's a very simple Raji tumor or a B-cell lymphoma tumor model. We did subcutaneous injection of Raji cells into the mouse and used rituximab to treat the mouse 2 weeks after tumor injection. And then only in mice with robust NK cell development, you can observe this ADCC effect mediated tumor growth inhibition, both in tumor volume and tumor weight. And we also find an interesting phenotype that before, after therapy, the blood NK cell look very similar in these SRG mice, but in tumor cells, we see much less CD16 expressing NK cells, probably either due to downregulation of CD16 or loss of CD16 expressing cells. But if we look into the tumor cells, tumor NK cells, what we see is that the 16 positive cells upregulate PD-1, whereas the CD16 negative cells do not. So that implies that the loss of function or exhaustion or death of CD16 positive NK cells. Then we did a combinational treatment. On top of rituximab, we also inject nivolumab, which is a PD-1 blockade antibody. And although rituximab alone already achieved a very significant anti-tumor activity, blocking PD-1 signal further reduced the tumor volume or tumor weight. Actually, when we look at these mice, we could barely see any tumor left. So it means that blocking NK cell exhaustion might be also helpful to boost ADCC activity. So whether the mouse model can be used for HIV latency, we have to actually do experiment. So we infect the mice, and then 6 weeks after infection. So normally in humanized mouse, the viral replication peaks within 4 to 6 weeks post-infection. And then at the peak infection level, we treat the mice with ART, a combination of 3 different drugs. And then the viral load drops to undetectable level about 4 weeks afterwards, and then remain undetectable. And our undetectable level is definitely not clinical sensitive level. It's about 100 to 200 copies per ml blood, but that's the lowest we can go. And if we stop our treatment, then the viral rebound. But the powerful tool of the humanized mouse is we can do a lot of operations. I don't have MD, so I decided to operate mice, which is totally legal. So you can take a mice, and after treatment, you can take this mice, which has at least 10 weeks of undetectable viral load, and then infuse a primary CD4P antibody by IV. So all the circulating CD4 T cells will be marked with red color. The tissue-redden memory cells, which have no access to the bloodstream, will remain unlabeled. And then you harvest the tissue and make single-cell suspension and stain them again with a different CD4 antibody. In this case, is a blue color. Then the CD4 T cells in the circulation will be double labeled. The true tissue-redden memory CD4 T cells will only be single labeled, which is here. And then with this method, we can actually separate circulating CD4 T cells versus tissue CD4 T cells, which is a nice tool because for patient, we normally sample in blood reservoirs. So we don't really have a powerful tool to sample tissue-reddened CD4 T cells. And even sometimes we get tissue biopsy, we probably still get contamination from the blood reservoir. So it's hard to distinguish them, and it's hard to tell whether they're different. Although we don't have any data regarding the viral sequence, but in this case, we're able to see HIV DNA in both circulating and tissue-redden memory CD4 T cell fractions. And the frequency is very comparable. If this is true in patient, then we can think about the fact that there are a lot more tissue-redden memory CD4 T cells than the CD4 T cells in the bloodstream. So then the relative contribution to total viral reservoir is probably a lot bigger in the tissue than in the circulation. So we definitely want to further investigate whether the viral sequence are the same or not, and how do they respond to anti-latency reversal agent as well as immunotherapy approaches. And we also did the same or very similar approach to look at the myeloid reservoir. So it's not surprising that in the circulation, there are only monocytes. So after 10 weeks of undetectable viral load, we didn't see any HIV DNA in the circulation compartment, which are short-lived monocytes. But we were able to detect HIV DNA in a tissue macrophage. In this case, this is the alveolar macrophage in the lung, so we were able to detect them. So this will give us some general idea that maybe this could be a model we can look at or modeling HIV latent infection or persistent infection in the myeloid compartment. And we can do a lot of further studies to characterize and develop therapeutic strategies against this particular compartment. So I have already briefly mentioned that it is very important to generate a patient-derived humanized mouse that reconstitute patient immune cells. It is important because eventually it gives us a possibility to test CAR T therapy, to test T cell engineering, or in vitro primed adoptive transfer T cell approach, or antibody, therapeutic antibody approaches. So our model can support hematopoiesis of the human CD34 positive cells. So this was a work done in collaboration with Bob Ehrlich's group, with the help from Christine Durand, to get HIV patient bone marrow CD34 cells, and we use these cells to engraft our mice. And 6 to 8 weeks later, we can see nice reconstitution of these patient-specific human CD45 positive cells, which includes monocytes and T cells, both CD4 and CD8. And then at the same time, we collect blood samples from the same patient to outgrow virus from resting CD4 T cells. So these are autologous virus, as well as CD8 T cells, so we can define peptide priming to boost antigen-specific CD8 T cells, and then adoptively transfer those CD8 T cells to the mice, reconstitute with autologous immune system, and infect it with autologous virus. So this whole system is the autologous system. And then in this group of mice, they either receive no CD8 injection or CD8 primed with the immunodominant peptide. As I mentioned, this peptide, vast majority of latent HIV carries KIT mutations within this peptide, so that the primed CD8 T cell clones will not be able to recognize them. So in this case, you don't see any inhibition of viral replication. But in these later two groups, either CD8 T cells primed by subdominant CTL clones or primed with a mix of different GAP peptides were able to fully suppress or partially suppress viral replication by measuring the plasma HIV RNA levels. And to look at the residual DNA, viral DNA, we look at the blood HIV DNA level. Then again, in this group, without injection of functioning CD8 T cells, you don't see inhibition or reduction. And in these two groups, we see that in some of the animal, there was a complete clearance of residual HIV DNA, or in other animals, there was a very significant level of reduction in HIV DNA frequency in the blood. So hopefully I've convinced you that this is the very useful humanized mouse models for HIV cure research, starting from the reservoir that we have humanized cytokines, including TPO, IL-3, IL-6, to boost early hematopoiesis and differentiate further into multi-lineage of human immune cells. And for the myeloid compartment, MCSF, GM-CSF are both important for monocytes maturation and further differentiation into tissue macrophages. And more importantly, for CD4 T cells, we have the IL-7 humanization to help the thymocytes development, as well as both IL-7 and IL-15 to maintain its long-term survival and a homeostatic proliferation of CD4 T cells. And we also have nice reconstitution of all the effector cells, which again, triggered by MCSF, GM-CSF, to develop monocytes, tissue macrophages. We also have IL-15 to boost NK cell response. In addition, we also have HLA-A2, B2M knock-in on top of IL-15 to boost CD8 T cell function. I have to point out that the B cell development in any given humanized mouse model is very underdeveloped, largely because we do produce B cells in humanized mouse. Actually, B cells is the very dominant population in most of the conventional humanized model. But those B cells are IgM or IgD single poly B cells because they cannot undergo class switch and somatic hypermutation process due to the lack of germinal center formation, which is follicular dendritic cell dependent. And FDC is a mesenchymal-derived cells. So in humanized mouse, we never put a human mesenchymal stem cells into the system. So, and mouse FDC cannot do the job. So we don't study IgG response in humanized mouse because simply they don't develop very efficiently into IgG positive B cells. There are some, but even with the very rare fraction of IgG positive human B cells in humanized, the mutation rate is also very low, still due to the lack of germinal center formation. So luckily, we can still utilize all the different kinds of broadly neutralization antibodies to test immunotherapy strategies in this case. So I would like to conclude and thank all the lab members at the Washington University in St. Louis, and also particularly thank to my postdoc mentor, Richard Flovell, who has invested tremendous effort and resource to make these humanized mouse models, which took 12 years. And all the postdocs who had contributed to the development of this model. And my PhD mentor, Bob Ehrlich, who not only teach me how to do HIV research, but also show me how to focus on very important topics that can have implications to clinic practice. And also show me how to stay focused on the very important question that you identify. And also, there are a lot of people being very helpful during my PhD training, including Janet, Kai, Jun, and Joe was very helpful in my CD8 T cell research. And Kristen is always helpful regarding patient recruitment, study design. And then Dr. Margolic group and Hao helped me a lot with the sorting. And also other collaborators and industrial collaborators, Regeneron, who co-developed the humanized mouse system with Richard Lab. And thanks for the funding source. Thanks.
Thanks so much. That was an outstanding talk. I think we maybe have time for one question before the coffee break. Bob or Trona?
Hi, Liang. That was incredible. I'm so blown away. I was wondering if you've done any of these CD8 transfer studies and looked at, in terms of whether tissue resident versus the circulating reservoir cells were preferentially reduced.
So I have never done that, but I think it will be interesting actually to further genetically modify. So we can, it's more like a missile. So once you prime CD8, you want to express the right chemokine receptors to trigger the T cells migrate into the tissue you want. For example, if you want to go to B cell follicular, you can express CXCR5. So, but I've never done that, but this is definitely doable in humanized mouse system.
Okay.
Yeah. Great. Maybe in the interest of time, we may wrap things up. We have a short coffee break, and then we'll come back with the primate models. Thank you so much.